In this video, we're going to talk about microbial growth. And as you may recall from previous videos, we said that microbes were ambiguous, meaning that they're going to grow everywhere. So they're going to be in that air we breathe. They're going to be in the foods we eat. They're already in and on our bodies. And ultimately, our goal is to learn how to control microbial growth in order to prevent diseases. In order to understand how to control them, we first need to understand what are those requirements that they need to grow. And those requirements can be both physical and environmental chemical needs for nutrients. And these are going to vary based on the microbe we're referring to. Now, again, we need to remember what the definition for microbial growth is. In microbiology, this is an essential concept. When we refer to microbial growth, we're referring to an increase in number of microbes themselves. So now let's dive in just a little bit into those essential factors, starting with the nutrients. So when we talk about nutrition, the term, that's the process for which the microbe is able to obtain the nutrients from its environment. Now we do divide or categorize our nutrients into either macronutrients or micronutrients, and that division is going to be based upon how much of it is needed. So it's a macronutrient if the organism requires a large amount and it plays an essential role in cell structure and metabolism. It's a micronutrient if it's needed in smaller trace amounts, and then those are going to be used for processes such as like enzymatic functions. In addition to understanding the terms for nutrients, we need to understand how microbes get those nutrients for their energy sources. So some of our microbes are going to obtain energy through chemical reactions. And if they use chemical reactions, we say they are chemotroph. Others are going to capture light energy for their energy source. And if they do that, we say that they are phototrophs. We also break down nutrients by how they get their particular carbon source. Is it going to be an inorganic carbon source or an organic carbon source? If it's inorganic, we say that the microbes are autotroph. If it is organic, we say it is a heterotroph. Another term that we use when referring to how microbes get their nutrients is, do the nutrients come from living or dead organisms? They're saprophytic if they are getting their organic material from dead host, or they're parasitic if they have to have a living host and utilize that tissue and fluids from that living, again, organism. Now, taking a step back just for a minute before we get out of the nutrients, we said that there were macronutrients, and the macronutrients were those that were required in large amounts. So there are 6 that we want to discuss: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. For each of the 6, you need to know what is the percentage of dry weight that is going to make up of the cell, and then why are they important. So a couple of examples of how the cell uses those particular elements for survival. So let's start with the biggest and probably the most obvious, that is carbon. Carbon is going to make up about 50% of the dry weight of a cell, and it's going to be used in most every structural and metabolic process that a cell has. So it is a backbone of organic molecules such as the lipids and the proteins, and as you may recall from previous discussions, these are essential for survival. The next is nitrogen. Nitrogen makes up 14% of the dry weight of the cell, and it's going to be essential in forming amino acids, which are needed for proteins and for the genetic materials within the cell. And they're going to get these from nitrogen gas or from nitrates. Oxygen. Oxygen is another essential macro element. It makes up about 20% of the cell weight, and it's going to be essential for structural and chemical processes, as particular enzymatic functions. Now, depending on the environment, will depend on how the microbe gets its oxygen. It may get it from inorganic salts, or if it lives in an oxygenated environment, it may get it from the oxygen gas itself. Hydrogen is another essential macro element. It makes up about 8% of the dry weight of a cell, and it's going to be necessary for maintaining pH and for forming hydrogen bonds, which is essential for water, and it's going to be required for a lot of other chemical reactions within the cell. So it is going to get hydrogen from organic compounds, from water, and even potentially hydrogen gas. Sulfur, it's a macro element as well, even though it only makes up about 1% of the dry weight of a cell, and it is going to be necessary for proteins and vitamin functions within the cells. And then our last one is the phosphorus. It makes up about 3% of the dry weight of the cells. Phosphorus is very important because of phospholipid for DNA and RNA, and in particular for ATP. The P stands for phosphorus, and remember that is our energy source. So remember the six, know what percentage they make up of the cell, and examples of why the cell may need these, what structural components, energy sources, etc. So now let's look at some of those physical requirements for growth, and we're going to break them down into the temperature, oxygen, pH, and osmolarity. So first let's talk about temperature, because temperature plays a pivotal role in microbial growth. Different microbes are going to thrive at different temperatures, and so we need to understand the terminology that we use for showing the preference or the optimal temperatures. Now, for bacteria in particular, they're going to have a minimum temperature, an optimal temperature, and a maximum temperature for growth, and we divide those into three groups based on their optimal temperature growth. So we have the Psychrophilics, which are the cold-loving bacteria. They grow best at temperatures between -5 and 15 degrees Celsius. Now we will be talking in Celsius here. And then those can be further divided into the true Psychrophilics or the Psychotrophs, depending on where that true optimal is. Is it below the 20 degrees Celsius or the 20 to 30 degrees Celsius? Then we have the Mesophilics, meso meaning middle. So these are going to be the bacteria that grow best at that moderate temperature between 25 and 35 degrees Celsius, even potentially up to 40, or maybe even a little higher, but their optimal temperature for many of these is 37 degrees Celsius, and hence that is body temperature. So this is an important group to remember. Then we got thermophilics. These are the heat-loving because they take it above the mesophilics, and where they kind of end at that 40, 45 range, that's where these start. They like their temperatures best between 45 degrees Celsius and 70 degrees, all the way up there. Most of them are going to prefer 50 to 60. Next, we're going to look a little bit at oxygen requirements, and this can be a little bit more complicated than the others. Oxygen requirements are going to be vital for microbes, whereas in others it could be deadly if you're talking about the environment. Okay, so here when I say oxygen, it's not the element oxygen that we were referring to with the macro elements and that it was essential for that. Here we're talking about oxygen as an oxygen gas. And again, as we said, microbes will vary depending on their species. So some are going to need oxygen and others are not. So when we look, we kind of break down and we term our microbes based on their oxygen requirement. At our highest level, you're going to find what we call the aerobic microbes, and these are going to be oxygen-loving microorganisms. They're going to thrive in environments which are rich in oxygen, and that's where they're going to perform respiration. They're going to take organic matter, they're going to convert it over into energy using oxygen. So ultimately we have what we call the obligate aerobic, where they're going to require the oxygen to live. We will then have, if you can look from those that like oxygen, we go down to what is known as the microaerophilics, and they're going to require oxygen as well. But unlike those obligate aerobes, which require oxygen for all things, they are going to only tolerate oxygen at lower concentrations because they're sensitive to oxygen, so too much of it can be toxic. We then have what we refer to as the facultative anaerobes. Now, the facultative anaerobes, they're going to be versatile because they're going to possess that ability to adapt to the varying oxygen levels. So when oxygen is abundant, they're going to use aerobic respiration and using the oxygen available. If it's there, they're going to use it. On the other hand, in the absence of oxygen, they're just going to switch over to an anaerobic metabolism using alternate pathways to survive. So our facultative anaerobes, they're going to be just very versatile. Do you have oxygen? They're going to use it. If you don't, they switch and use the anaerobic processes. Then we have our obligate anaerobes. And these are going to be those microorganisms which are anaerobic specialists, if you will. What do I mean by that? That they are unable to tolerate a small trace even of oxygen. So they're going to flourish in those environments which have no oxygen at all, and they're going to use anaerobic processes to survive and even fermentation abilities. Okay, and so then we get to our last one, which is going to be the aerotolerant anaerobes, and they don't use oxygen at all for any of their metabolic processes, but they are able to tolerate environments which do have oxygen. Why? Because they're able to still break it down and prevent it from being toxic to them. So if you look, this is in the notes and stuff, kind of at a growth pattern in a test tube. This kind of helps explain sometimes how those or those oxygen terms and what they mean. So imagine if the test tube is open, and so therefore you have air coming in. So you can see the growth patterns are wherever it is yellow, that means there's growth in microbes. So here, if you have the oxygen coming in, now let's also remember about oxygen's levels as they move through fluids. Okay, oxygen can diffuse and dissolve in a aqueous solution, but the farther you get down in the environment, that water environment, the less and less oxygen, and it can only go to a certain level. So here there's going to be high levels of oxygen, and down by in here you're going to have no oxygen. So that should help you kind of understand these growth patterns. So in A here, we have high levels of oxygen, and that is the only place you see microbial growth, because that's going to be the ones that are going to be obligate aerobes. They have to have oxygen. And where I'm getting these terms from are just back up here, okay? Microaerophilics is the next one. So notice if this is high level of oxygen here, so right here you notice there's no growth, but as we get into lower levels of oxygen, that is where these thrive, because they need oxygen but only in smaller amounts. Now see here, this is the facultative anaerobe. Remember those that are real diverse and they can live with or without oxygen. They just adjust their metabolic processes. So they love to be in oxygenated environments, but can still thrive when the oxygen level decreases or goes down to essentially none. D is going to be the obligate anaerobes. So again, you notice here that you're going to have the ability to grow where there is no oxygen, but it doesn't grow beyond that. And then in E, we have our the aerotolerant microbes, and they can grow wherever. They don't use oxygen. They have methods to help eliminate the toxic forms of oxygen, so they can grow pretty evenly at any level of oxygen. So again, these test tube pictures here are just to kind of help you understand the terms and the definitions put right here. Okay, so let's move on to pH. This is a little easier. Most of you are very familiar with pH and the fact of the pH scale of being 0 to 14. And if it's below 7, it's acidophilic, and if it's above 7, it's going to be in the basic alkaline groups. Well, when we're using the terms to describe microbial growth and their optimal pH temperature, as you can see here, the terms are very self-explanatory, which is nice. So they're acidophilic if those microbes like it in a lower pH, and generally below 7 to the point that they like it at about 5.5 or below. Neutrophilic microbes like it right there in the center, somewhere between 5.4 to 8.5, where alkaphilics love that alkaline basic environment. So they're going to like it when it's above 7. Particularly, most of them will have an optimal between that 9 and above. So again, these are very self-explanatory terms. One other thing that we have to look at when we are talking about our bacteria and their growth patterns and where they can live is we need to look at the osmolarity. Okay, and osmolarity is going to be a little bit different of a concept, but what I need you to recognize is when we talk about osmolarity, it is how much water versus how much salts or osmotic pressure are found in a solution. So this is a crucial role in determining if something is going to survive or not. You will see this concept is also going to come up because we exploit this when we're talking about food preservations and things like that when we get to the control part of the unit. So first, let's talk just a little bit here. Let me orient you to the pictures. So here you see that we have cells. So in each of the three definitions, the top cell is a normal, thriving cell that lives in optimal conditions. So what happens with the bottom picture then is what happens when we put it into an aqueous solution that is either isotonic, hypertonic, or hypertonic. So again, let's just start by giving those definitions. Something is going to be isotonic when that osmotic pressure, okay, and again, when we say osmotic pressure, what we're simply referring to is how much particles, how much salts, how much whatever it may be that's dissolved, those solute particles are dissolved within an aqueous solution. Okay, so when we say isotonic, what we're saying here is that the surrounding environment is equal to the inside or the cytoplasm of the cell. So if the solution that you take that cell and you place it into this solution, if it has equal water, equal salts both on the inside and the outside of the cell, then any water that moves in and out is going to be equal, and the cell's going to thrive in this scenario. Now, that is ideal, but sometimes cells get into environments that may have more solute and less water, or less solutes and more water than where they currently survive, okay, or currently live. So in this case, let's talk about a hypertonic solution. If it is a hypertonic solution, then the osmotic pressure is high. And what that means is that you have a lot of solutes, a lot of particles in the aqueous solution. So your solutes go up, whereas your amount of water or the solute goes down. So when you do that, one of the things that we need to understand for hypertonic and hypotonic, so the next two, is that water flows from an area of high to low, so it goes downhill. So what we're saying in these diagrams here is that the cell has high water, low solutes, and we're putting it into a solution that now has high solutes and low water. So if the cell has more water than its outside environment, look what happens to the water. It pulls out because it's going to go from high concentration inside the cell to the low concentration out here in the environment. So if you take a cell out of its normal environment and you put it into a hypertonic solution, the cell is going to lose water and literally it's going to start to dry up. Now you know that because a high salt solution would be considered a hypertonic solution. And what do you know about if you put something in a high salt solution? It's used for drying. And why do we dry things? Because we're pulling the water out. Now, the opposite occurs in a hypotonic solution. In a hypotonic solution, the osmotic pressure is low, and that means that there is more water and less solute in the solution than what's in the cell. So here's our cell in its ideal condition, and now we just put it into a solution that has greater water out here and less solutes, and in the cell now has less water. So if you have less water in the cell, more water out of the cell, again, water goes from high to low. So if you've got high outside, low inside, then water is going to come in. Now, what's going to happen as you start moving water into the cell? The cell's going to expand and expand and expand. And in the case of a bacterial cell, for instance, it's got a cell wall, so it's going to allow it to expand some. But if it's too much water coming in, ultimately it's going to be like a balloon that you put too much air in. It's going to pop and it's going to explode. So in hypertonic means high water. Now again, this is a concept you already recognize. Let's say that you're in the kitchen and you have celery. You've done, or you may have done this in lab. It's a little bit wilted. What do you do to make it crisp again? You put it into a glass or bowl with fresh 100% water, and then that allows the water to go back into the celery cells, pump them back up, and the celery becomes crisp again. It's that same concept here with hypotonic. Now, why does all of this work and everything? It's because of the salt levels and that play a role in this. Okay, now when I say salt and when we say osmolarity, it's not always salt, but salt is a big reason that we talk about this. And so put some terms down here just to help you understand. So halophilic, so halos are the halogens, those are the salts. So we do classify our bacteria in particular on their ability to live in high salt concentrations or not. So if we say that it's a halophilic bacteria, that means that bacteria has to have a larger concentration of salt, such as like the ocean water. So it's going to take in have a lot different process and levels than something that might be might be like a bacteria that lives in our skin, because we're not going to have that high level of salt. If we say an obligate halophilics, they require very high, so 20 to 30% salt. And facultative halophilics or bacteria do not require high salts but can tolerate it up to about 2%. So if they're halophilic bacteria, then you have to kind of adjust what amount of osmolarity will cause the bacteria to either thrive or to be killed. And last, let's talk about just a little bit of when we talk about bacterial growth and microbial growth. Not only is it, as we said, those physical and some of the chemical characteristics, but also the environmental. So for this, all it is learning the definitions. That is all you need to worry about for this. So microbes, for instance, can thrive in certain situations, but not in others, based on the organism or the environment. So we say in some terminology here, if it's symbiotic relationship, then that means the organisms live in close proximity with the other organisms in that environment. So if it's a mutualistic environment, then that means that the microbe and the organism that it's living with are going to both benefit from that relationship. If it's a commensal, then that means that one organism is going to benefit and the other is just maintaining. It doesn't necessarily benefit, but it's also not necessarily harmed. Whereas if it's a parasitic relationship, then one organism is going to benefit and the other is going to be harmed. And we often refer to a lot of our microbial relationships as being parasitic to their host. And you might also recall we used the term parasitic earlier for nutrients, how a microbe gains its nutrients, because it has to be living tissues if it's parasitic. So in this case, the microbe would be the parasite and the host is going to be the one harmed. So those are symbiotic relationships and where they live in close partnership. It's either going to benefit them both, one will benefit, the other is neutral, or one is beneficial in gaining benefit and the other is harmed. Then we can have non-symbiotic relationships where the organisms are free living and don't necessarily rely on each other for survival. But if it's a synergistic relationship, both of those organisms living in the same environment, they're going to benefit and share nutrients independent of one another, but they're going to both survive. If it's an antagonistic relationship, then they're going to be in the same environment and they're going to compete against one another for those nutrients. So that is the end of understanding about microbial growth and what they need to be successfully growth. Next, we're going to talk about how we use those factors, exploit them in order to control their growth.